we've spent the last year, or more than the last year, pretty much obsessing about COVID-19. I don't think that's exclusive to people that have been working on COVID-19, but in my group of people, we've been pretty much exclusively thinking about that and very little else. So I'm going to talk to you a little bit about a short whistle-stop tour of some of the things that we've done, and hopefully attract some nice discussions with people in the rest of the school and beyond. So please do email me if there's any questions. There's my email. I've just joined, very happy to be here. Okay. So we spend a lot of time thinking about the way that behavior or human behavior affects epidemics. We've been doing this for some time, and this year has been the time where we've actually used it in battle. This kind of knowledge in battle, to try and mitigate some of the effects of the COVID-19 pandemic and answer some policy questions that have arisen throughout the year. So I sit on some government panels, the sub-panels of the SAGE Scientific Committee, that regularly get thrown questions about what we know and what we should be doing to stop bits of the pandemic going on. And these are just some of the questions that we've been asked throughout the year. There's not an exhaustive list, and we didn't get to answer all of them very quickly, and I'm certainly not going to be able to cover them in 5 minutes. But I thought I'd just pick out some of the key ones that got us involved, and that have a very sort of strong data or mathematical modeling component, which I hope is going to be of some interest here. So at the very beginning of the epidemic, we wanted to know how many people could be infected. That was one of the first things that we were thinking about. That's, I don't know if you could, I'm hoping you can see my cursor, which is pointing right at the beginning, which is where we sort of started thinking about modeling the COVID-19 pandemic in the UK. And we did some work based on work we did for flu back in 2009, rapidly developed our flu model, which looked a bit like this, and that was pretty scary. We adapted that to COVID-19 rather quickly, probably in the space of about a week, to take the basic structure of the model and change the parameters, the epidemiological parameters, in these sort of standard compartmental models to take in COVID-19 parameters as they were being estimated, again, very quickly from the outbreak in China. The reproduction number, one of the key parameters for flu, is about 1.5, for SARS-CoV-2 is about 3, and that has massive implications for how quickly and how much the disease is going to spread. We made estimates about the other parameters also rather quickly, probably in the first 2 months of the pandemic, with our colleague Rob Challen, who was at Exeter at the time, but is also hopefully joining us here in the next couple of months at Bristol. Okay. We also regularly and still do estimate the reproduction number in the UK from all sorts of data sources. And hopefully try and refine how we do these estimates on a regular basis, but it remains a challenge, particularly as the data sets keep changing. We do spatial estimates of R as well, which also keep changing, particularly in light of new variants. Okay. These are some of our early predictions that we made. We made a guess about what seasonality was going to do, but it turned out that behavioral changes at the beginning of the epidemic were the things that were really driving the epidemic down and forcing a second peak past the summer. And very quickly, I think I'm going to run out of time very soon, but I just wanted to raise another of the questions that we tried to answer throughout the pandemic, and what is the impact of variants as they arise. So the variants of concern, the Kent variant appeared sort of late in the winter, or became a concern in December or so. So we set out to measure the epidemiological impact of this using the databases that we were getting from the well, through the government channels essentially, and found that there was spatially the reproduction number was higher where there was a lot of variant. So there was some there's some geographical correlation between the two, and ultimately we did a large kind of data intensive study on the mortality of the variant and found that it was much higher. So here is the key figure, which is a survival curve. For the new variant, the survival curve is lower than for the old variants, and this implies that the new variant is substantially more deadly, unfortunately. This is something that we continue to do with new variants, and if anybody, there's some very nice mathematical problems that we haven't yet solved, so if anybody would like to come and help us, please do get in touch. This is just a very quick sketch. I'm happy to share these slides and have a conversation with anyone who'd like to get involved in some of the work that's still going on and is still very important for our response going forward. I'm going to stop there and just thank all my funders and people that have helped.